Hey all, it's Dr. Anderson. So for those of you that are still struggling a little bit with these Hardy-Weinberg problems, I thought I'd throw together another little example to help walk you through that you can look through a time or two if you are still confused on how to work these problems out. So in our example here, assume that you're a fisheries biologist and you are working with a population of fish that has two strikingly different morphs. So there is the blue morph, and there is also the rainbow morph. And from past research, you know that the blue morph is dominant over the rainbow morph. So blue morph dominant, rainbow morph recessive. So you have been studying these fish in a few different lakes, and in one of your study lakes, you've sampled 80 fish. And what you find that of those 80 fish, 65 are of the rainbow morph. So if you know that 65 of your fish are of the rainbow morph, what then does it tell you about how many fish you have in the blue morph? You should be able to figure that out pretty quickly in your head and deduce that you have 15 of the blue morph fish in your population. So now back to our problem. We want to know what the frequency of the dominant allele is, how many heterozygous individuals are in our population, and also how many homozygous individuals are in our population. So we'll shrink these guys down and think a little bit about this. So first things first, we have to think about those two equations that are associated with the Hardy-Weinberg equilibrium model. You can pause the video now and think about what those are, write them down, and think about what each means. But the first equation, that simple one, P plus Q equals 1. And then that big mouthful of an equation, P squared plus 2PQ plus Q squared equals 1. So we got our two equations here. So what do those two equations mean? What do they represent? Well, our top equation represents phenotype, frequency, whereas our bottom equation represents then genotype, frequency. So we'll just keep these in the background here for later reference. So next let's recall each of the parts of those equations. So we have our P, we have our Q, we have our P squared, we have our Q squared, and we have our 2PQ. So all the parts in the equations above we have here, and just a quick recall of what they mean. Again, our P is the frequency of the dominant allele. Q is the frequency of the recessive allele. P squared then is the frequency, just put F for frequency, of the homozygous dominant individual. Q squared is the frequency of the homozygous recessive individual. And then 2PQ is the frequency of the heterozygotes. So we'll put this up here for safekeeping as well. So now we're actually ready to start solving our equations. So the question is, what do we know? Which of these pieces of the puzzle, which ones do we know given our information? So we'll throw up each of those components we need to solve for. Going back to our problem, we again remember we know that of our 80 fish, we know that 65 are of these rainbow morphs. So those rainbow morphs, again, we know are, from the problem, we know that they are recessive. So if we know that 65 of 80 are recessive, which of these pieces here do we know? So think about that for a second. Should come to the conclusion that the piece that we know is this guy right here, q squared. And this is the part where a lot of students get a little bit confused. Some will go for this guy here, but it is q squared because they are recessive, therefore they have two recessive alleles, or diploid individuals. So q squared we know to be 65 out of 80, and that is equal to 0.81. So given q squared, what's the next piece we can find out? Well, another place where people trip up is thinking that now that we know q squared, we can solve for p squared by simply subtracting q squared from p squared, but that doesn't quite solve our problem, as p squared, those dominant individuals, could be homozygous dominant, or they could be heterozygous dominant. So they could be either here or they could be here. We don't know. Just because you are a dominant individual does not mean that you are homozygous dominant. You could be heterozygous dominant and have one recessive allele locked up inside. So given q squared, we can then solve for, what we can do is solve for q. So we can do that simply by taking the square root of 0.81, pull out your handy dandy calculator and do that, and you should get a q squared of, or a q rather, of 0.9. Now that we know our q, we can simply solve for p, and all we have to know to do that is this equation here. So we solve for p1 minus 0.9 equals our p, and that gives us a p of 0.1. And from here on out, the rest is pretty simple, just throwing the values into our equation. So 0.1 squared equals 0.01. 2pq, so solving there, 2 times 0.1 times 0.9. Do the math there, we get 0.18. So the last thing is just to go back to our original question, make sure we've answered everything. First part, what is the frequency of the dominant allele? That is this guy right here. Next part, how many heterozygote individuals are in our population? Well, we know the frequency of heterozygous individuals is right here. So if we simply multiply our frequency by the total number of individuals, so by 80, we will come up with our answer, which will give us an answer of 14.4 individuals. Well, we can't have a portion of an individual, so we'd probably just round down to 14. So that would be our answer for the second part. So last, homozygous dominant individuals, same thing, multiplied by the total number, multiply the frequency by the total number, and we get 0.8. Again, we can't have a portion of an individual, so round up to about 1, and that would be our answer there. So hopefully this has been helpful, walking you through each of the steps, getting a better grasp of how to solve these Hardy-Weinberg problems.